Good morning, ladies and gentlemen. It's my pleasure to give you a short introduction to a technology that will allow you to do tissue cytometry at super resolution. I am actually a adjunct professor at Queensland University of Technology in Brisbane, where I lead a group working on AI-based solutions and AI-based decision support systems for precision diagnostics. But today I speak to you in my role as co-founder and CEO of TissueGnostics. The company has been founded in 2003, so next year we will celebrate our 20th anniversary. The headquarters is located in Vienna, in Austria, and we have a network of international subsidiaries, offices, and distributors on all continents. The company offers a set of slide scanning instruments with subsequent image cytometry software that we call TissueFAXS. So in analogy to the flow cytometer, the FACS, we bring this idea to the slide. The instruments are available in different configurations for transmission microscopy, fluorescence microscopy, spinning disc confocals, or to do whole slide confocal imaging, also an inverted setup where you can do live cell imaging and time kinetic experiments, a high throughput configuration with a slide loader for 120 slides, and a spectral imaging system using a liquid crystal tunable filter. You may recognize, or you will for sure recognize, that the hardware is based on a high-end microscope from Carl Zeiss, Germany. So we are an OEM partner of Carl Zeiss. And what makes TissueGnostics, or what converts the Zeiss microscope into a tissue cytometer, that is our software. Both software that automates the scanning, but also the software that does the analysis. We have three applications. They're called HistoQuest, TissueQuest, and StrataQuest, with StrataQuest being our high-end solution that allows to do contextual tissue cytometry, that has deep learning functionality, machine learning module, and allows to do high-end solutions in image cytometry. You can really understand it or read the result, the readout, like in a FACS plot, just that one dot, or each dot here that you see in the scattergrams relates to a slide and to the cell on a slide or in a tissue section. There is a very nice functionality that links the two, the image with the data set, so you can click on an image and the corresponding object will start blinking in the scattergram. Following our motto, from image to data, we consider this a step forward and thus call it the forward connection. It also works the other way around. You can make a gate, which is a selection of a subpopulation of cells, and the content of the gate will be highlighted in the image. So you have an immediate link between a cellular phenotype, a subpopulation of cells, and the corresponding localization in the sample, so histology. We can do whole slide confocal imaging, as I mentioned. We see here an example from one of our clients, Janilia Farm. This is a whole mouse brain section. To acquire it at 20x in 4 color channels in a single plane, it takes 8.5 minutes. Each of the rectangles that you see here are one high power field of view. 350 fields of view have to be acquired, each one with overlap to each other, and then they are pixel-wise matched, which is called image stitching. If you acquire also a set stack of 21 layers, so main focal plane, 10 layers above, 10 layers below, the scanning time increases to 1.5 hours, but you acquire essentially almost 30,000 images and 115 gigabytes of data. And then you can navigate through the sample, like in Google Earth. Here you will always see the map that shows the entire sample, and the red rectangle here indicates what you see in the main window, and you can zoom in to see the detail down to the subcellular details. Now, and I would really like to emphasize that this is whole slide imaging, not just one high power field of view. Again, from another client in Jacobs University in Bremen, a whole mouse brain section, and when we zoom in here on this, what is called third ventricle, you can see the high resolution details. Now we can combine this technology with another technology that we have been introduced to yesterday in the opening session by Ed Boyden, expansion microscopy. So I can keep this short. Rather than introducing ever more technically demanding and also more expanding solutions, Ed and his team had the ingenious idea to reproduce the expansion of the universe on the nanoscale. We all know that resolution in optical system is defined by the ability to resolve two dots that are close to each other. What expansion microscopy is about is basically like inflating a balloon. When you inflate it, two dots close to each other get further apart. So they introduce a scaffold that can be then expanded. And this we have done here on as an example on a prostate section. We can see the prostatic glands. This is the overview. When we zoom in, we start to see the details. This is 25%, 50%, this is 100%. The tissue has been expanded 4 times. This is a 40x objective lens. And what we see here exceeds the resolution of, for example, a confocal using a 63x, because 4 times expansion with a 40x magnification gives you a virtual magnification of 160x. And what we can see here are the incredibly detailed structures, even of the chromatin inside the individual nuclei. These are is the cytoplasm of the secretory epithelial cells, the lumen of a single prostatic gland. And the white dots here are individual vesicles, individual secretory events that we can visualize here. And bringing these two technologies, TissueCytometry and expansion microscopy together, this is what we call expansion-based super resolution.